every day in the mirror I see a woman. And every time I look in mama, I see that woman. A wonderful woman, an African woman, mother of nature. Beautiful woman. Beautiful woman. May I buy, baby, ni buy. Semiga sensual yensha, neyenza. Me was super wild, but you're my one vet to say. Baby, ni buy, baby, ni buy.
Hello, everyone. Thank you for making time to join us today. We'll start very shortly. Hi. Thank you, Jeffrey. That's fine. Hello, everyone. Thank you for making time to join us today. I must say that it's going to be a really exciting time. Today, we'll be diving into an exciting intersection of science and maternal health. We are going to be looking at preeclampsia genomics. Lots to learn, lots to share. And yeah, I'm really excited about this. So we want to be on time, so we'll just get right into it. I'll be hosting today with Jeffrey behind the scenes. And so pardon me, we will be running through some preliminary stuff before we actually go into the presentations. Next slide, please. All right, so before we begin, kindly make sure that your microphone is muted. If you want to ask a question, go for it in the chat. We'll be monitoring the chat from time to time, and we'll make sure that we bring your questions to the attention of our speakers. But please make sure that you're on mute so that we can have a good time together and we can hear everything that is being said. In case we want you to ask your question yourself, we invite you to do that, and you may need to unmute to ask your question. Please note that we record all of our webinars and they are made available on the APEC Ghana website and our social media platforms. If you don't want your name or your video to be visible, please go ahead and turn your video off, or perhaps make your name anonymous, but you're also welcome to keep as is. Next slide, please. All right, so before we begin, we just have a few opening exercises, and then we'll hear from one very brave survivor, and then we'll introduce our speakers and they will present, and we'll have a Q&A session. Next slide, please. Great. So where are you joining us from? If you can please type in the chat, where are you joining us from? We want to know what town, region, or country you're joining us from. I am joining you today from Abuja, Nigeria. So I'm originally based in Australia, but I'm in Abuja this week, and it's very hot here. So that's where I'm joining you from. So Johnson is also joining from Ghana. Charity from Ghana, you're welcome. I see someone from Buenos Aires. Akuradi. Oh, wow, lots of Ghanaians, really impressive. And someone from Lithuania, Samuel, you're welcome. Dar es Salaam, that's where Lusago is joining us from. Thank you for making time to be with us. We have lots of people from the Volta region, from Kumasi. Thank you for making time to join the APEC webinar. Okay, Kingsley says Kumasi, Edwina says Ghana. Where exactly in Ghana, Edwina? But thank you for making time to be with us today. So keep the chat going. Just type where you're joining us from. And you can also include your profession. So I'm a midwife, and I'm currently a midwifery researcher. And so we want to know more about you. So Camilla says she's a cardiologist. Amma is joining from Oti region. Welcome, Amma. Thank you for joining. Soraya says she's joining from Sunyani and she's a midwife. So we have lots of midwives. We have obstetricians, Asin Fosu. Great. Thank you for joining us all the way from every corner in Ghana. We are really excited that you could make time to be with us. As I mentioned earlier, there's a lot to learn. The word itself, genomics, is like it's quite big. So if you can put in the chat, what are you hoping to learn today? It can be a fun fact. You can also decide to put a fun fact about genomics in the chat, or any relatable analogy about genomics. That would be interesting. You can put that in the chat as well. But if you just want to share a sentence about what you hope to take away from this session, go ahead and put it in the chat. All right, Prince, thank you for joining. So Prince is a medical officer. Darling is also a medical officer from Koforidua. Thank you for joining us. We are also live on Facebook. So you can go onto the APEC Ghana page and you can share with all your friends and colleagues on Facebook. All right, so thank you. At least I've gotten to see where some of you are joining from Sugakope, from Kasoa. Really interesting. Thank you so much. So Lisago says he's looking forward to knowing more about the genes that are associated with preeclampsia and any new updates. So am I, Lisago. I'm looking forward to hearing more about genes and about preeclampsia. Well, let me share one fun fact that I read about genomes. So I read somewhere that although our genomes are 99.9% identical, it is the 0.1% difference that makes each of us unique. Really interesting. So think of genomes as a whole menu of options or
Isabella, it looks like you're breaking a lot. Could you check your connection? Well, whilst Isabella is trying to reconnect, we will continue the meeting. I think she shared a very interesting fact about how those 0.1% is what makes us all unique. So let's keep remembering that. Well, the theme of the overall Knowledge Cafe has always been to assemble healthcare professionals and other associated caregivers, researchers, and the lot to under the field of maternal health care delivery, and with that also improve the knowledge and improve the care for women at risk of HDP or who have HDP or have survived from HDP. Now, genetics in preeclampsia is an important factor. I'm sure a lot of people keep thinking about whether this condition runs in their family, whether there's a genetic predisposition, and what can they do about it. So the more we look into our knowledge and the more we gather knowledge about the genetics behind preeclampsia, the better we can improve the care for our women. So we expect with such Knowledge Cafe sessions, and particularly with this topic, that clinicians and other members of the scientific community will understand the role of genetics in preeclampsia, and with that be able to have a better look into the predisposing genetic factors and molecular pathways involved. We also hope that with this knowledge cafes and the information we gather regarding genetics, that clinicians and others will gain insight into how the genetic findings can improve the management and treatment of preeclampsia, and to also use this knowledge to address gaps that may exist in research and potential areas for future research on genetics. Well, most important now is we want to listen to one story. Let's see what the impact of the knowledge we have and the research we do has on our patients. So I would like to invite our dear sister Ofusua Koilabi, if she's online, to briefly share her story. Koiwa, are you there?
Yes, I'm here. I understand that we may have to unmute our participant, so I'm trying to find her. She was online a short while ago. I'm not sure.
Okay, that's fine. Maybe we'll just go ahead and it will make
Okay.
time for her when she comes back. And sorry about that. My internet just snapped and I went off, but I'm back.
Hello, please, I'm here.
All right,
Okay,
great.
great.
Over to you,
Okay,
Ofusua.
thank you. Thank you very much. So my name is Ofusua Koilabi, and I'm here to share my story. So I actually am very much aware of preeclampsia because of APEC Ghana. And so in the beginning of my journey as a pregnant woman, sorry, I decided to, take all the precautions, which is I tried to eat healthy as much as possible and reduce my level of salt intake. And I tried to monitor my BP every now and then. Yes. So everything was fine. My BP was actually very normal, right, from first trimester to the ending of my second trimester. It was usually around 120/80. And then when I entered my 30th week, um I noticed that my face started swelling. Yeah. And I started experiencing headaches. The headache wasn't so serious. It was intermittent. So I used to just take paracetamol for it. And then I decided to, report to my doctor. And I think on the 30th week, I went for my antenatal visits, and then there was when they did my urine tests, there was +1 protein, and my BP had gone to 130/90. Yes, so the doctor put me on Nifecard XL, but when I started taking Nifecard, the headache even got worse. So I reported to him, and then he switched me to methyl-dopa. So I think the good thing that helped me was because I'm also a health professional. So I was always communicating with my doctor. And like at any point in time, when I feel unwell, at any point, I let him know. And so I made him aware that the Nifecard was, I don't know, I just made him aware that I wasn't seeing any improvements because the headache was becoming worse. My face was swelling even more, and my leg even got swollen at a point. My left leg got swollen at a point. And initially, I had to be admitted at the hospital. They did some tests, and um, they actually diagnosed me of having DVT, deep vein thrombosis. And I was wondering where that was coming from. But then upon subsequent tests that was run, I mean, there was no DVT. So clearly, I felt like there was some sort of medical negligence over there. And so the swelling reduced on its own. I was admitted on the hospital. I was put on TED stockings and then obviously elevation of legs for some time. So I was just admitted for like 2 days, and then I was discharged with the leg coming back to normal. This was in my 30th week, so my early stages of third trimester. And then when I noticed that the headache was getting worse, the doctor decided to change the Nifecard XL for me, and then he placed me on methyl dopa. And that was in my 33rd week because I was going for the antenatal every 2 weeks when I got to my third trimester. So the 33rd week was when they switched to methyl-dopa for me. And the doctor also gave me IM dexamethasone. He said that he's going to let one of the midwives inject it for me, so that in case of anything, they will be able to do, in case of any emergency, they'll be able to do a CS or, all of that. So in my 33rd week, I started taking the methyl-dopa, and then I was also going to hospital to get injected for the dexamethasone. Yeah. And then I think on the day that I got my first shot, my first dose of dexamethasone, I started feeling tightness in my thighs. And because I was new to pregnancy and all of that, I had no idea. I just recently found out that tightness in the thighs is also a form of contraction. I had no idea. So I just knew that it was like, it's as if someone was holding my thighs, and it was just like the person was gripping it. And as I was explaining it to everyone in my house, nobody could understand me. So that evening, I had to tell my husband to take me to the hospital no matter what, because it kept getting worse. And then I got to the hospital. I was admitted through the emergency unit, and then later they referred me to the obs and gynae ward. So at the emergency units, they monitored the fetal heart rate, and it was normal. But that was Friday evening. And then at dawn, the nurse came back to monitor again, and she realized that the fetal heart rate was coming down. So she immediately called my doctor, and then they liaised together, and he came in and told me that we had to do an emergency CS. So I did an emergency CS Saturday morning at 9 a.m. And by God's grace, the baby came out healthy, was taken to the NICU. But after that, my BP shot up. The tightness in my thighs became worse. My legs got swollen, and like the BP it kept shooting up. So I was in the hospital for like 2 weeks. I was being put on. I was still taking the methyl-dopa after the emergency CS, but it wasn't doing anything. So later, they had to put me on labetalol, and they kept increasing the dose. Labetalol tablets, they kept increasing the dose, but even with that, the BP was not stabilizing. Like, it was always around 168, 114, 168, 114. There was a time where it shot up to 198, 114, and then after that, I think the doctor decided to do the labetalol IV. I mean, their whole protocol, and then MAC sulfate, and then it came down for a while. So it came down to like 150, 110. So at that point, I was switched back to the labetalol tablets. I was being put on it, for some time, and I was still in the hospital being monitored and all of that. And then it became stable around 140, 90. Even with that, the doctor was a bit skeptical in discharging me. So he had to monitor me for an extra day or two. And then we noticed that it came down to 135 over 80, 85 to 90 days. So it was hovering within those ranges. And then I was discharged on that. So I was discharged on labetalol tablets, 400 milligram every 12 hours. And as I kept going for my postnatal reviews, the BP was coming down, so he kept reducing it gradually. And currently, I am on labetalol 100 milligram once daily. So that's pretty much about my story. Thank you.
Thank you so much, Ofosua, for sharing your story with us and giving us a glimpse into your first-hand experience of preeclampsia. And this just underscores how serious preeclampsia is and how we need research that is going to make things a little bit easier for moms who develop preeclampsia. Because imagine if a single genomic test could tailor your pregnancy care plan just for you. How would that change your outlook on pregnancy care in general? Thank you so much for sharing that with us. Next slide, please. So my name is Isabella Gatti, and as I mentioned earlier on, I'm a midwife. I'm a midwifery researcher based at Charles Darwin University in Australia, and I'm your moderator for today's APEC Cafe, together with Dr. Jeffrey Ahin in the Netherlands, and he's also my co-moderator. So thank you so much for joining us here. We will be engaging you in the chat. So please keep your comments coming, keep your questions coming, and we'll do our best to bring them to the attention of the speakers. The next slide, please. So we've come to the fun part. This is why we all logged on to know more about preeclampsia and genomes. So I'd like to first go ahead and introduce our first speaker, Dr. Lily Pemka. So Dr. Lily Pemka is a molecular geneticist and senior lecturer at the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. She is also the Deputy Director of Research and Entrepreneurship at the West African Genetic Medicine Center in Accra, Ghana. So Dr. Lily Pemka, over to you.
Hi, thanks. Let me share my slides. Can you hear me clearly?
Yes,
Yes,
please, we
we
can
can.
hear you.
Okay, all right. Okay, good. I now see the window. Let me start to share. It's not giving the option to share. Okay, let me look at the screen.
You're able to share now?
Yeah, it's showing, let's see, it's not sharing. When I try to share, the window that comes up
Hold
doesn't
on.
include my presentation. Okay, let me close it and start again.
If you're having difficulty, we could assist. Jeff?
Okay, let's see.
Yes, I made the options available for you to share, Lily.
Okay. There's nothing showing my window. It
Jeff, you
allows
can-
you to share, but it doesn't, nothing pops up.
No, otherwise, I can share your presentation if you give me a second.
I'll have to give you the new one because I made some changes to it. Let's see, why not? I can send you right now because when I try to share, it just doesn't show up on that window, in that window. So
Whilst we wait to get this sorted, guys, just type in the chat. Those of you joining us now, thank you for joining us. You're welcome. And we want to know where you're joining us from, what you do, and what you hope to get out of this session. So let's keep it interactive. Go into the chat and type where you're joining us from, what you do, and what you hope to get out of this session. I'm going to read a couple from those who just joined us. So Daniel is saying hello. Gifty is also saying hello. And Soraya is asking a question about Ofosuwa's story, asking about pain in the thigh from the injection.
Jeffrey,
Yeah.
I sent it to you right now.
Yep, great.
All right. Thank you, John, for joining us. So John is a physician assistant joining from Akentin Health Center. I hope I said that right. And Camilla says I'm a cardiologist and I'm joining
Jeffrey,
from...
sorry, let me interrupt. Sorry to interrupt. Jeffrey, how do I send it to you? I'm not able to, because nothing shows on my screen.
Please send it over to Koiwa, and that should be fine.
All right, guys. So thank you for joining us from Nkoranza, West Municipal Hospital. Joycelyn, thank you for joining. Hilda says, I'm a midwife and I'm joining from Tamale. That's nice. Florence is a nurse manager joining from Dix Cove. Nancy is joining from Cape Coast. Eunice is joining from Tyne District Hospital. Great to have you. Gifty is a midwife, also joining from Pramso. Someone is joining from Konfanochi and says wants to know more about preeclampsia. Then we also have Daniel, who is joining from, well, I can't see where Daniel is joining from. Then Charles is a physician assistant from Donko Chrome. Thank you for joining us and making time to be with us today. We are just sorting out a few technical issues and we'll kick the ball rolling. Sharon says she's from Accra. Rebecca from Presbyterian Health Center, Enchi. Thank you for joining us and keep your comments coming. And what do you hope to get out of this session? When we say genomes, what comes to mind? If you can just type in the chat, that would be great. So Hannah from Accra, Dr. Techi from Kwanta, thank you for making time to join us. Yamfo Health Center, thank you guys for joining. OUB Health Center, great. Ikropon Health Center, thank you so much for the interest and thank you for joining. This is by far a very big audience for this webinar. We are so glad that there's such interest. And yeah, we can't wait to hear what Dr. Lily Pemka has to say. Lily, are you ready so that we can allow you to have your presentation? Amrahia Health Center, Suhum Government Hospital. This is like the whole of Ghana. Really impressive. Bawe, SDA Hospital, Bichem. Wow, thank you for making time to join us. Lots of people from Tain, District Hospital, Tema Polyclinic. Great. Albert is a physician assistant from Winti, all the way from Winti, and wants to know more about preeclampsia and the genetics of it. Ivy is from the University of Ghana Medical Center. Jesse is from Tema Polyclinic, Rara Government Hospital. Wow, that's really impressive. Selena says she's from Kaneshi Polyclinic. Esther is from the Trust Hospital. Thank you for joining us. Just a few technical issues with internet and all of that, but we are ready to go. All right, over to you, Dr. Pemka.
All right, thanks. So like I was saying, she rightly introduced me. I'm a molecular geneticist, and I study genetic traits and diseases focused on cancer. Of no, I don't necessarily work on preeclampsia, but I have a project where my PhD student who just submitted worked on spontaneous preterm birth. So interesting thing is one of our exclusion criteria was to exclude women with preeclampsia because we wanted to focus on the spontaneous preterm birth and try to be as focused as possible on the traits we're focusing on. So with talks like this, I like to do a bit of some basic genetics so we can understand, first of all, the topic and understand what else we're going to discuss today. So this is the outline. I'll talk about the human genome. I'll talk about complex and Mendelian traits, touch on preeclampsia, and then we'll talk about genetics, epigenetics, a few other topics, and then we'll conclude. So what's a Human Genome Project? So it's one of the most audacious projects that was conducted in the world, basically. But let's start off by looking at the human body. The human body is made up of cells, and in the human cell, you have the nucleus where you have the chromosomes. In humans, in diploid cells, you have 46 chromosomes, but in the egg and sperm will have half of that, which makes sense because after fertilization, you want to have a fetus that has 46 chromosomes. Now, when you look at the structure of the chromosome, so this is a condensed chromosome, is actually made up of all these, the DNA, as well as certain proteins like histones. And these are proteins that the DNA is wrapped around. So if you look at the DNA, it's a double helix, and it's wrapped around these histones, which are then tightly packed into into chromosomes, which are now kept in the nucleus of the cell. Now, the basic unit of the DNA is what we call nucleotides. There are 4 types, A, T, G, and C, and they are arranged in a linear manner, and they form a double helix like that. So these are the nucleotides I just mentioned. Now, people wonder how the genome works. Now, it gives instructions on how an organism develops. Now, how the instructions are given are based on the DNA code. Now, what happens to the DNA code is it's transcribed. One string of it is transcribed into what we call RNA, an intermediate form. And then the instructions are read by the ribosome, which reads it by looking at 3 nucleotides at a time. And those 3 nucleotides at a time, each code for an amino acid. So when it reads the code, it keeps translating the RNA into proteins. And then your protein is a string of amino acids. So we have about 20 amino acids, and then you have a string of protein. So this is basically what it has. It's central dogma of biology. It's not strictly, it's not strictly adhered to, because there are times where you can have different activities, like where you have RNA that are actually functional, you know, and then you have the end products being proteins on this end. Now, this is just basically a sequence of the nucleotides, and just for brevity, you have a really short sequence. And in the human genome, I mean, you have about 2.5% is actually what you have, actually genes. The rest of our regulatory elements, etc. So we'll make this really short. So for example, in the DNA, this is a DNA sequence. I mentioned the nucleotides, so they are arranged in a string-like form. And then what happens is you have sections of the DNA that are actually genes. So imagine the ones that have the blue outlines are the genes. And each one would be transcribed into what we call the RNA, the intermediate form, and then will now be translated into proteins. And these proteins are involved in so many things. And this is what you see here. So these are just like 3 genes, just summarized for you to understand. Now, what's the role of the human genome? It's basically the blueprint. I mean, it's like when you build a house or any building, you need a blueprint, you need a plan, and that's what you adhere to build the house. Now, it basically has all the instructions necessary for development, function, and reproduction, and it explains variation. I mean, when you look at people, no 2 people are identical, only identical twins. And the reason why this is the case is because variation in our DNA, those sequences of the nucleotides vary a bit from one person to another, okay? Now, it helps us understand human biology, how it works, how we grow, how we metabolize things, and then how we even respond to disease. And so it basically gives you the genetic basis of traits, your height, different things, and diseases as well. Now, the moderator mentioned something about being 99.9 identical. When you look at the nucleotides and compare one person to another, we're 99.9% identical, but that tiny percentage is what causes the variation. And most of it is, there are so many things in the genome that, you know, account for the variation. And one of the major ones are called SNPs. And they're called single nucleotide polymorphisms. And basically, I'm talking about a single nucleotide changes in a single nucleotide. So these are just variations of basically flavors. So if you take 3 people, for example, and you align a particular gene, for example, you see that people have different nucleotides at that point. And a lot of times, these polymorphisms don't necessarily contribute to disease. They contribute to variations. But occasionally, some of these rare events are what we call mutations, and those can actually cause disease. Now, we can't talk genomics without the Human Genome Project. This is the most audacious biological project that was ever conducted. It started in 1999. The first draft came out in 2003. And that was the first time the human genome was sequenced. Honestly, the reason I'm a human geneticist, and I mentioned anytime I talk about the human genome, was because I took an advanced genetic course in level 400. And the lecturer was talking about the Human Genome Project. The draft came out later that year, but I was talking about the plans to do it. You're talking about 20 labs and 13 countries around the world, it took them 13 years to sequence a human genome for the first time, and it cost $2.7 billion. And now today I can get a genome sequence in hours, and to cost me a few $100s. Now, from the Human Genome Project, before that, we didn't even know how many genes we had in our genome because we have a massive genome. And now, based on that, we're able to show we had 3.3 billion base pairs, like 3.1 billion base pairs, meaning the number of the nucleotides I mentioned in one cell. So this, if you say haploid, so I'm talking about half of it. So you're talking about what you have in the egg or in the sperm. So if you pick a diploid cell, for example, what you have in your skin, you have 6.4 billion of those bases. And we have an estimated 20 to 25,000 genes. So what this Human Genome Project did was provide fundamental information about the human blueprint. Even then, as a student, I could appreciate the impact of something like that, but I couldn't imagine what the Human Genome Project has done to transform the world of genomics. And when you talk genomics, you're talking about the entire genomic makeup, all the genes in the entirety in an organism. So following the Genome Project, a lot of things came up. Now we have next generation sequencing. Before that, you had Sanger sequencing. That sequence only 1,000 base pairs, for example. Imagine 1,000 base pairs when you have 6.4 billion bases. So you could only sequence short fragments. But with the next generation, with the Human Genome Project, it provided the full sequence. So now next generation sequencing, what it does is it chops out the DNA, sequences it. Now you have a framework where you can easily map it up using bioinformatics. And the word sequencing, I know a lot of people became familiar with it during COVID, is basically sequencing or determining the sequence of nucleotides in the genome, and that's sequencing. Now, what's happening is, hold on one sec. So what is done now is with the Human Genome Project is giving rise to an era known as the omics era, where you're able to see the entirety of genetic makeup of any organism or any cell. So other than genomics, which is looking at the DNA sequence, you have epigenomics. It's basically so people, when people talk about genetics, they forget it's what we call epigenetics, right? And epigenetics is another level. It's above genetics that regulates how these genes are expressed, basically how these genes make their products. So for example, if you take a human being, you have over 200 types of cells, but every cell is genetically identical. Why are the cells different? Your skin cell is markedly different in function and phenotype from a nerve cell, for example. It's because there are certain genes that are only made in certain tissues. They're called tissue-specific genes, and those are the ones that give the cells the phenotype. Although for all the cells, they produce certain, they express certain genes that are called housekeeping genes, and like the name suggests, to keep the cell, going basically. So that's one of the major mechanisms that regulates whether certain genes will be expressed in one tissue versus the other. It's called epigenetics, and that's another thing that you can look at. You have transcriptomics, basically looking at all the RNA, which is the intermediate of DNA in the cell. Now you have protein, and that's when you're looking at the entirety of the protein, and that's proteomics. So I'm touching on this because in the topic talks about genomics and all this other omics. And metabolomics is where you analyze all the output from all these pathways and reactions in a cell, smaller molecules like amino acids and whatnot. It gives you an idea of what's happening in the cell, what is being broken down, what is being metabolized. Now, we talk about genes, the environment, and traits. I know I was talking about genes, but there are certain situations where something is genetic, but you don't quite able to determine the pattern. With sickle cell, we know it's recessive. you can tell from a pattern that, okay, this person has to get 2 copies of the mutant allele to show the phenotype. For some of them, it's dominant. The person only needs one, something like Huntington's mutation, Huntington's disease. However, there are certain genetic diseases, you know, it's running a family. The pattern is not quite clear, but you know it's genetic. So on that note, it brings me to talking about genes and the environment. Now, few genes act alone. You don't have, most of the time mention, oh, this gene is involved in this disease. It's not just working in isolation. It's working in the pathways, interacting with other genes and so on and so forth. Now, other than other genes that can modify the expression or the function of one gene, the environmental factors as well, and they can modify it. So certain traits, when you look at traits, it can be grouped broadly into Mendelian diseases. Like you have sickle cell. It's one gene that's involved. You either have it or you don't. And they have polygenic. That is caused by multiple genes, like your height. The genes for height are several genes will determine your height. Now, in these situations, they can also be multifactorial, meaning that this gene is interacting with the environment, and that's where you can introduce complex genetic traits. Now, when you talk about complex traits or multifactorial, talking about genes and the environment, things like height, skin color, weight, blood pressure, and all these things, cancer susceptibility, all are complex genetic traits. It's not just the genes, but the environment is involved. For example, look at skin color. Other than the genes, even exposure to UV light can modify what it looks like. Height, it's influenced by genes, but depending on how you affect growing up, you can be stunted or not. And that's the environmental factor. Even the way you respond to disease, for example, COVID, there are certain genes that have been identified to predispose certain people to more severe COVID than others, like the blood group system. And then finally, preeclampsia, which is what you call a complex genetic trait. Now, what's preeclampsia? It's basically high blood pressure and organ dysfunction during pregnancy. It's the leading cause of maternal and maternal mortality, 3 to 5%. In other situations, you are seeing up to 8%. And so the main symptoms are here: severe headaches, which the survivor mentioned, vision blurriness and light sensitivity, abdominal pain, breathing difficulties, and also swelling of the face and extremities. That's quite a common phenotype, sudden weight gain, sudden and rapid weight gain. Now, it's basically, putting it simply, it's a combination of immune regulation that's disrupted, or then vascular dysfunction between the fetus and the placenta. So together, it leads to abnormal placental development and the other symptoms that are associated with preeclampsia. So some of the risk factors, a few of them, you have obesity, hypertension, diabetes, and then of course, family history, which I'm going to focus on. Usually when you talk about family history, it's usually an indication of a familial component, because what happens is, I mentioned that, you know, we are 99.9% identical, but however, the more related you are, the closer you are related, the more likely you are likely to share some of these SNPs. And so family studies have shown that if you have like a first-degree relative, you're likely to, you know, your risk for preeclampsia goes up. And there have been studies where they've identified genes from both the mother as well as the fetus that contribute to developing preeclampsia. So there are key maternal genes that are affecting immune regulation. So of course, when you're pregnant, your immune system is kind of dampened, and there are genes that regulate that. So when there are certain variations in that compromise that, you know, activity, then it can contribute to that. So talking about vascular function, clearly, when you look at the placenta, there's a lot of vascularization that connects the fetus to the mother. And so if there are variants in genes that regulate that, that can also contribute to that. You have fetal contributions that I already mentioned. For example, certain variations, you know, the fetus comes with, of course, your genes, half of your genes, and then the father's genes. So there are certain genes that are expressed in the fetus that would regulate activities like this and contribute to preeclampsia. Now, with the advent of genomics, there's a tool that we use that's called genome-wide association studies. Basically, what happens is, especially when dealing with traits where you want to find if there's a genetic component, what you do is you stratify your group, those with the disease and those without, right? And then you genotype them, basically just looking at the SNPs. And what happens is, so in a situation like this, you have people with, say, preeclampsia and women who did not develop preeclampsia. And then you now genotype their SNPs. And what you do all these association studies. And what it does is it now helps you identify SNPs that are more common in those with, in the patients. And when you find those SNPs, it gives you an area in the genome that points to genes that could be regulating that disease you are dealing with. The same thing was done with COVID. We were able to find genes that are involved in immune regulation and so on and so forth. So that's, in this studies like this, you talk about thousands and thousands of people. So I give an example of a recent study that was done by Jaco et al. In 2023. A GWAS study was done for those with preeclampsia and other hypertensive pregnancy-related conditions. So this is the Manhattan plot I showed you. I'm not going to bore you the details, but basically it shows you we have 23 pairs of chromosomes, right? So it basically looks at each chromosome, and then what it does is, and now you compare those who had preeclampsia versus others. These guys looked at about 17,000 pregnant women. And then what it does is, like I mentioned, it's going to highlight certain areas in the genome that are likely to be associated with it. So you have the preeclampsia phenotypes, and it picks all these genes at the top here. So it should reach a certain level of significance. It's really stringent, looking at using some statistical tools. And if you look at preeclampsia or other maternal hypertensive disorder, you realize they have similar genes with some others that actually overlap. It tells you that, hypertension, which we mentioned earlier, is actually linked to, preeclampsia. Now, from this genome-wide association study, what they noticed was they had 19 genome-wide significant associations. So there were 19 areas in the genome that lit up. And out of this 19, 13 were novel, meaning that 6 of the genes have already been seen. And that's usually significant and then reassuring, especially it confirms certain studies you've done in the past. If you're able to see other loci that have already been identified, loci mean locus, a location in the genome. 13 were novel. 7 of the new ones were genes that were previously associated with blood pressure traits. So they've been identified with blood pressure, but here we identified and then in relation to preeclampsia. And these are the genes that we know they're associated with blood pressure traits. So if these are genes, like I mentioned, your genes are your blueprint. It basically regulates practically everything about you, including, the ones that are just Mendelian and the ones that interact, are complex, interact with the environment. So it was really significant for these genes to be highlighted. Now, they also identified 4 genes involved in the development of the placenta. Now, the placenta is heavily involved in the disease you are dealing with. So imagine you have genes that have been implicated in the development of placenta with variations. That tells you, well, those genes actually make sense. So that's one of the things you need to focus on when you do studies like this. You find so many genes. Where are these genes located? What are these genes known to be? What are the function of the genes? And once they fit in, they actually make sense. And then following that, you can conduct all these other functional studies to figure out what these mutations or variations are, how are they contributing to disease. Now you're talking about uterine spiral arteries, which are heavily involved in the pathogenesis of preeclampsia. They identified 4 of them. Kidney function. You talk about increased protein in your urine, which is one of the key signs of preeclampsia. And they found genes that are involved in kidney function. And then you have maintenance of proteases. proteostasis. Talk about protein in the urine. So basically maintaining that protein balance, that protein balance in the pregnancy serum. It has to be at a certain level. When you are above, you are excreting protein in your urine of a certain, above a certain amount. Clearly, it's pathogenic. So these were the genes, which makes absolute sense. And so in a lot of situations, we are dealing with some of these complex traits, like I mentioned earlier, you can have multiple genes, it will be multiple. Some might have variations in all the genes, and they contribute. So you have gene-gene interactions, as well as the environment. Maybe the person was overweight and all these other things that the person is exposed to that can now contribute. The food, the diet, the salt, all those things now contribute to pathogenesis of it. You know, I mentioned epigenetic factors. You know, a lot of times we do genetic studies, people do genetic studies and exclude genetic factors. So one of the interesting things is these epigenetic changes that I mentioned, it doesn't affect the actual genetic, the DNA sequence. It affects one, there's something called methylation, where there are certain marks, methyl groups that are placed on cytosine, one of the nucleotides. And usually when it's placed in a certain way, mutation in the gene sequence itself, it can silence the gene. So sometimes you do a study, you want to check if there are mutations in the DNA, you don't find any mutations. So the assumption is that that gene is fine, so it's something else. No, it's not always the situation. When you look at the epigenome, you do epigenetic studies, you're able to see that actually, in actual fact, this gene is methylated and they're highly methylated. So it's not even making its product. So essentially, it's also silent. So those are some of the things you look, and I mentioned that the histones that are the DNA is wrapped around. So the histones are also regulated in that manner, can be acetylated, methylated, and a lot of times it also contributes to whether the gene is open, that region, euchromatin, or is packed, that's heterochromatin. So you have the DNA regulatory machinery, or DNA what do you call it, transcriptional machinery, not getting in there to turn on the gene. So, and epigenetic changes can actually be influenced by the environment. And so things like toxins, their diet, exercise, etc., can modify some of these things, and it can lead to so many different traits, including disease susceptibility or behavior longevity, including what do you call it, preeclampsia. So there are certain situations where, so I have a project in my lab, my student works on prostate cancer, and we did epigenetics, and we basically looked at the epigenome, and we found certain genes that are known to be mutated at the DNA level that predispose people to prostate cancer, now being methylated. All the sequences, it looks fine, but it's actually silent. So the epigenetic factors that are actually being also being implicated. And there's also what we call microRNAs. I'll get to it. So based on these findings that people have conducted, sometimes it's a single gene they look at, it's called that genetic study. Can look at a whole genome where they do what we call whole genome. You sequence the entire genome, the 6.4 billion bases, or you sequence just the exome, the regions that actually have the parts of the genes that make products. It's like 1% of that. It's not the entire gene, the whole gene, the gene takes about 2.5%. And then they look at they have all the genomic studies. So based on that, all the findings that have been done, there are some key insights that make sense, that show the molecular underpinning of preeclampsia. For example, you mentioned immune dysregulation. There are certain proteins that need to be expressed that will help the mother tolerate, you know, having a foreign, in quotes, fetus growing here. If there is disrupted, that can contribute to preeclampsia. Abnormal.
Hi, Doc. Sorry to interrupt, but can you please round up in 5? We'll come back to you.
Yeah, I'll be done in like 5 minutes.
Thank you so much.
Normal placental development clearly can contribute to that. Oxidative stress, when it comes to mopping up oxidative stress, that can cause cell damage and so on and so forth. And mutations in that can contribute to that. MicroRNAs also are under epigenetics, and they regulate, you know, gene expression by preventing translation of the mRNA. And then you have all these interaction with the maternal, with the mother and the fetus that can also contribute to that. Now, what are the applications and what we learned from that? It can help with genetic screening, because if you know some of these genes that can predispose you to preeclampsia, if you're able to detect them, then you take certain precautions that will prevent you from developing that. Then there's personalized medicine. The whole world is moving towards this area because now we are genetically we are different. And if we have, we're able to now personalize someone's medicine based on your genetic makeup, even the way you metabolize drugs, that's pharmacogenetics, it helps. Now therapeutic targets, you understand the genes involved clearly, and then also the pathways that underpin it, you can now target it. And then with current research, we're able to see, first of all, it's a complex genetic architecture. And one thing that we need to point out is the fact that there's limited studies in more diverse population. If you look at Black women, for example, Blacks in general, genomic studies, there's only 2.4%. 78% are in white people, Caucasian populations. But clearly, when you look at genetic traits, they present with racial variations, just like looking at how we look physically is underpinned by genetics. So clearly with genetic diseases, it's really important to study the population to understand all the peculiarities that are happening, that are the peculiarities of the molecular underpinning of the disease. Black women are more likely to get it. They are more likely to, and one, because the incidence of hypertension, obesity, and diabetes is actually higher. So if these are the risk factors, it makes sense. And then of course, our genetics, we are not clear on that, but that's a gap that we need to address. The next speaker has done a bit of a study and he's going to talk on that. And so in conclusion, understanding genetic contributions is key to combating preeclampsia. And if you're dealing with a different population, it's really crucial that you study that population so you can understand, the nuances, the genetic differences that are driving that phenotype in that population. All right, thank you.
Thank you so much, Dr. Premka. That was really elaborate, and yeah, we've picked up a few questions we'll ask at the end of the second speaker's presentation. But the little I can pick up is that genomic research can really help us identify like genetic markers that signal if this woman is at increased risk of preeclampsia or not. Thank you so much. So we'll just move on to the next speaker. We have Dr. Claire Simpson, and she's a statistical geneticist, that's a big word, and a genetic epidemiologist and associate professor of genetics, genomics, and informatics at the University of Tennessee Health Center in Memphis, Tennessee. So Dr. Claire Simpson, over to you.
Hello. How do I make the slides advance?
Sorry, you can just give me a note when you want to move on to the next slide.
Okay. Well, thanks everyone. I'm going to sort of follow on from Dr. Premka's talk to talk about a specific gene in preeclampsia. Next slide. So I'm going to talk about 4 pieces. I'm going to talk about the APOL1 gene, specifically how it relates to preeclampsia, how we've been studying that gene in the SPOT study, and the outcomes that we have so far. Next slide. So to start with the APOL1 gene and its evolutionary history. Next slide. So human African trypanosomiasis, also known as African sleeping sickness, is a disease, a parasitic disease called by Trypanosoma brucei, and that is transmitted by the tsetse fly. There are 2 types in humans, T. brucei gambians and T. brucei rhodesians. There are about 70 million people at risk. There's about 11,000 people currently infected. We see 2,800 new infections each year. In 2015, there were 3,500 deaths, which is a lot less than it was in 1990 at 34,000 deaths, but still a very highly deadly disease. Next slide, please. Yep. So there are variants in the APOL1 gene that are reasonably high frequency in African populations, and those variants arose in high frequency because they offer protection to the individual against trypanosomiasis. So if you have the baseline allele that we call G0, you do not have any protection from HAT. If you have one of the alleles, the G1 or the G2 alleles, then you have what we call heterozygous advantage. That means you are now resistant to HAT. But if you have two of these alleles in any of the three combinations, G1, 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 G2, or G2, G2, then that's also a homozygous disadvantage. You are resistant to HAT still, but you're also at risk of chronic kidney disease later in life. And the reason this works out this way is because trypanosomiasis obviously is deadly and may well kill you as a child before you reach reproductive age. Whereas chronic kidney disease is something that happens in later life, long after you might well have produced children. So there's no selective pressure against the gene variants by the existence of kidney disease. Next slide, please. So the worldwide frequency distribution of these two variants, G1 and G2, is concentrated exclusively in Africa, specifically sub-Saharan Africa, and in African Americans whose heritage is primarily West African. Next slide, please. And so what's interesting is when they've drilled down into the risk models for these two alleles, that you see different responses depending on which one that you have. So for example, carrying one copy of the G1 allele decreases your risk of severe HAT. If you have two copies of G1, your risk of chronic kidney disease is up, but you still have that reduced risk of severe HAT. With the G2 model, having one copy of the G2 allele decreases your risk of infection by HAT, but does increase your risk of severe HAT if you were to contract the disease. If you have two copies of G2, you, in addition to those two risks profiles, you also have an increased risk of chronic kidney disease. Now, you'll see that if you look at the overlap between the distribution of the two subspecies of T. brucei, you'll see that there is a concentration of the APOL1 alleles in those areas where the parasites are most common. What's a little curious is that there's this quite high distribution of the G2 allele in West Africa, which is the allele that gives this other risk profile. And since the majority of the T. brucei in West Africa is T. gambians, it's a little strange that G2 is concentrated so much there. So it's something where really our understanding is still evolving as to how APOL1 responds to the risk of these two parasites. Next slide, please. So that's a little bit about APOL1. The question now is, what's the relationship between APOL1 and preeclampsia? Next slide. So what is the evidence we had for a role of APOL1 in preeclampsia? Well, we know that the placenta is one of the highest sites for APOL1 expression. We know that the APOL1 gene cluster is abnormally expressed in preeclampsia. We have seen transgenic mice that express APOL1 in placenta develop preeclampsia or eclampsia phenotype. And we know that pregnancy serum markers associated with preeclampsia risk include APOL1. Next slide. So here in Memphis, we undertook a look at this study called the CANDLE study. It was an observational cohort of 1,500 healthy pregnancies. And the study is still running. It's been running for about 10 years now. But the important thing is that of the 1,500 women and mom-baby pairs in the study, about 2/3 of them were African American. Now, yes, it was an observational study, but because of the high rate of preeclampsia in African American women, that's why some of the pregnancies that were healthy at recruitment went on to develop preeclampsia. Next slide, please. So we had our 990 African American mother-baby pairs, of which 921 mothers and 747 infants were successfully, the DNA was extracted and genotyped. Next slide, please. So what we found was that the association with preeclampsia by APOL1 genotype in our study, which is panel B, I'll explain panel A in a sec. In panel B there, you'll see the UTHRC study, the odds ratio or the effect size for the APOL1 high-risk genotype basically was double the risk compared to the baseline, whereas the maternal genotype did not seem to show a significant effect. And when I first did this study and I did the statistics, I was actually kind of worried because I was like, if I mix the fetal and the maternal up, I don't understand. Went on to talk to some placental biologists and they said, actually, yeah, this makes total sense. So we then collaborated with some folks at Albert Einstein College of Medicine. They saw a similar pattern of risk, as you can see in panel A. So yeah, so it's about a doubling of the risk of preeclampsia. Next slide, please. So we published this in 2018, showing that this result that this fetal and not maternal genotype of the APOL1 gene associated with this increased risk of preeclampsia. Next slide, please. So that's what we know about APOL1 and preeclampsia. The next thing I want to talk about is the SPOT study. Next slide, please. So one of the things that's sort of a fundamental thing is that with genetic results, like with most scientific results, you need to see replication. And so we needed to replicate in other cohorts. Unfortunately, within the US, there were no cohorts of African American with preeclampsia at all. Click there. And the few observational cohorts that were out there that had African Americans in them didn't ask about obstetric history at all. And those that did ask about obstetric history didn't have very many African Americans. So we were kind of frustrated. And so through a chain of acquaintances, we met Dr. Joyce Brown from the University of Utrecht, who, you know, she was performing a clinical trial of the Piers and mini-Piers instruments to determine if those scores could accurately predict which women were at highest risk. So we met her and we all agreed that another study could piggyback on some of the infrastructure she had built to do our replication study. Next slide, please. So this is how the study that we called SPOT Bio was conceived. We needed funding, and although we considered submitting a proposal to the National Institutes of Health here in the US, those of you in the audience who've been involved with these kinds of applications know that the process is very, very slow and uncertain. Next slide, please. Now, it just so happened that Dr. Davis knew someone at the Bill and Melinda Gates Foundation, and this foundation has historically been very invested in maternal and neonatal health in LMICs, and our contact was very enthusiastic about our ideas, so he invited us to write a proposal. Dr. Davis, Dr. Brown and I, in collaboration with Dr. Emmanuel Serfeño at the Greater Accra Regional Hospital, we came up with this general plan. We would aim to recruit 800 women who presented with preeclampsia and 1,600 controls at 26 to 34 weeks. And we chose this early onset preeclampsia because the disease can be more severe with early onset, and from a genetic point of view, is more likely to have attributable genetic risk factors. So we chose the Greater Accra Regional Hospital, Paula Boo Teaching Hospital, La General, Tema General Hospital, and the Eastern Regional Hospital in Cuff Ridgeway. Obviously, La ended up not being used very much because they knocked it down. Next slide, please. So we decided to use the same questionnaire that Dr. Brown was already using so that we could cross-compare the two cohorts. We consented the women for broad data sharing and asked if they would also consent to being followed up. We took a range of samples, maternal blood and urine at recruitment, and cord blood and placental tissue on the baby at delivery. Next slide, please. And to our considerable delight, the Gates Foundation agreed to fund us. Next slide, please. So, as you can see, we managed to recruit actually more cases than we had initially aimed to. There are some technical reasons for that. And we did manage to achieve a similar number of controls, although not as many as we originally planned, which had been double that amount. As you can see, I've marked it here, the COVID-19 pandemic really caused us some problems, and it meant that we started control recruitment much later than would have been ideal. Nevertheless, we did manage to collect a lot of samples, which makes this one of the biggest cohorts of preeclamptic women of African ancestry in the world. Next slide, please. So since we published our paper in 2018, there have actually been several other studies that sort of replicate our result. So, for example, you see the first two studies there are from our paper. There was a study in South Africa which only looked at moms and seemed to show a dominant mode of inheritance as opposed to the recessive mode of inheritance that we had seen. Equally, an Ohio study only had infants. They also showed a dominant effect. There was also a nested case control study in Boston, which showed varying results depending on the ancestry of the moms, whether they were Haitian or African American. But the issue with all of these studies is that they're all very small. The Ohio cohort is the biggest in terms of the number of cases, but the number of controls they have is very small still. And so when thinking about things like model of inheritance, these kind of small sizes make it very, very difficult to tease apart what's really going on. Nevertheless, there has been replication of our results, so that's encouraging. Next slide, please. Another thing that's come up since our 2018 paper is another variant in APOL1 that I'm going to call N264K. And this variant is co-occurs with the G2 allele. In some cases, you also see it on the G0 background. What you don't see is it in the G1 allele. And this variant protects patients with the G2 allele. If they also have this N264K, it protects against this this kidney disease. Next slide, please. And by inhibiting the APOL1 pore-forming function, which is how it disrupts the trypanosome, this just shows that it quite strongly prevents APOL1 mediated kidney disease. If you look at this forest plot, you can see that if you look at the red results in each of those two plots, you can see that having the APOL1 high risk genotype in the absence of N264K, you have this very strong association. But if you have N264K, even though you have G2, your risk is pretty much the same as if you were wild type, if you had the G0 allele. And that's, like I say, that's true in both chronic kidney disease and end-stage renal disease. Next slide, please. And another thing that's been going on is drug companies have been looking at small molecule APOL1 inhibitors as a precision medicine approach for APOL1 kidney disease. And on the screen here is just some of the molecules that they found. They obviously did some sort of broad small molecule screening approach to find these molecules. Next slide, please. And so they recently published this paper with this drug they call inaxaplin, sorry, and done clinical trials to see if this drug is effective for people with a high-risk genotype and kidney disease. And they're showing that there does seem to be some hope for this drug as a means of testing of treating kidney disease. Next slide, please. And so, as you can see, you can see this by eyeballing the bars on this bar graph. There's this very strong effect, even though the sample size was very small. There was only 30 people in this study. But you can see that the change in protein to creatinine ratio, which shows improvement in kidney function once the drug is started. And you can see that the impact, if you look at the bottom panel, you can see the impact appears to persist over time, which is obviously important for a chronic condition. Now, like I said, all of this work has been focused on kidney disease. But we think it might be interesting to see if these drugs perform well and their toxicity is low. The question would be, would these drugs also be effective in high-risk women, or rather women with high-risk fetuses, as a method of preventing or reducing the severity of preeclampsia in those women. Next slide, please. So moving forward, we are about to embark on the statistical analyses of our study, the Spot Bio study. Concurrently, we actually have two other studies ongoing looking at proteomics and metabolomics. And we're also always looking for future collaborations. Anyone who's interested to contact us, and we'd love to talk to you about what you're doing. I have to acknowledge all our partners and funders for this study, including the Ghana Health Service and Corlebury Teaching Hospital, H3 Africa, the Netherlands Organization for Scientific Research, the Bill and Melinda Gates Foundation, and my university, the University of Tennessee Health Science Center. Last slide, please. And this is the huge number of acknowledgements that I have to make. I can't call out everybody. But particular thanks go to Dr. Srafenyo, Dr. Brown, Dr. Adubon-Safo, Dr. Davis, Dr. Adu, who were all fundamental to this project. And thank you very much.
Thank you very much, Claire. And thank you very much, Dr. Pemka. We have received so much information, like this is an information overload. Thank you so much. in this short time, we have received a lot. And there are a couple of questions we'd like to go through. This question is for Dr. Pemka. There were lots of questions around the environmental causes when you were talking about the epigenetic factors, and many people are asking if you could explain that a bit more in a very simple way that they can understand. So just 2 to 3 minutes, Dr. Pemka. Is Dr. Pemka here? Okay, I think we...
Isabella, Dr. Pemka is having some technical issues, so
No
unless
worries. Then this
connection
question can be,
assist.
I want to ask another question to Claire.
I'm here.
So Claire,
Hello?
all right, Dr. Pemka, did
Yes,
you hear the question?
I did. I actually
All
need to
right.
if you're available, anyone has my number, could send me the link to the Zoom call because I need to leave, so I have to join on my phone so I can pick up my kid from school before 6. So yeah, I can go over some of the environmental factors. I mean, there are nutritional deficiencies. You can talk about stress, pollutants like heavy metals, stuff like that. Even lifestyle factors. If you're not eating well, I mean, poor lack of physical activity. Talk about infection. All those are environmental factors and stuff. So yeah, all other things that are not necessarily genetic could be actually in the environment. And so I don't know if you wanted me to touch on the epigenetics, how these things can also influence your epigenetic factors.
Yeah, just a bit.
Yes.
Thank you.
So with the epigenetic changes, I mentioned DNA methylation, histone modifications, and then the microRNAs. So these are things that can affect the way certain genes are expressed or even silenced, and without necessarily impacting the actual DNA sequence. So certain things in the environment, even in the diet, can cause certain genes to be methylated, and so they can now be silent, instead of being expressed. I hope you got that.
Yes, thank you very much,
Vice
Dr.
versa, and there are
Pemka.
certain things that could be in the environment that could also, instead of a gene being silent, or being underexpressed, it can cause it to be overexpressed. It can change the epigenome. So that's one part of it.
Thank you very much. And Claire, over to you. This question, someone is asking that what was the justification for choosing the study sites for the APOL1 study?
So partially it was because of these were the hospitals that Dr. Brown had been working with, so there was also already a relationship there. And then the additional hospitals that we added were just to expand our pool of potential recruitments as much as possible. And these hospitals were identified as having the highest birth rates, so that we had the best chance of getting as many women as we wanted.
All right, thank you so much, Claire. In fact, we have a lot of questions, but we've run out of time. So what we'll do is we'll answer the questions and then we will find a way to disseminate it, because we've really run out of time and we have to round up. Next slide, please. So great. What struck you the most during this webinar? If you can type in the chat. I have already typed my takeaways in the chat, but what are some of the things that you will remember? What are you taking away with you from this webinar? Lots of technical things about genes and genomes and DNA and ribosomes and all of that, but what are some of the things that you will remember, your greatest takeaways, or any new thing you have learned that is sort of like increasing your knowledge of preeclampsia and the pathways to which women develop preeclampsia? If you can type that in the chat, that would be nice. Thank you. And will you be joining us for the second Knowledge Cafe? So we have a series of Knowledge Cafes. This one was about the genomes and preeclampsia. We have one coming up on their metabolomics and all of that. So will you be joining us for the next webinar? Have you learned something? Do you want to come back for more? You can type in the chat, and yeah, I would love to hear from you. Yes, the recording will be available on our social media handles. We don't ordinarily share slides, but the recordings will be available. And so you can watch the recordings on YouTube, on Facebook, on our Facebook page, and you can feel free to share the link and to, share the link and also, interact with us some more on our social media pages. All right, so if you want to follow us, we're on Facebook, we're on Twitter, we're on Instagram, and we are also on WhatsApp. Subscribe to our newsletter, really informative. We have different sections in the newsletter tailored to everyone's needs, from the pregnant woman to the health provider. So kindly scan the QR code and have a look at our APEC newsletter. We have lots of editions with lots of informative stuff. So we will send the certificates for those of you who registered. If you want to join our WhatsApp community, also you can do that by following the link or scanning the code. And yeah, it's been great to have you for the last hour and a half. If you want to support us, kindly send us an email, or you can send us a WhatsApp, or you can actually make a bank payment or send us your donations through our Momo accounts. Our merchant number and details are displayed on the screen. Thank you guys so much. I'm seeing lots of very interesting comments in the chat. People say it was very informative and they'll be joining us again. Thank you to our planning committee, the CEO of APEC, Koiwa Koilabi, and the team. Thank you so much for holding the fort and, coming up with all these ideas for us to meet and learn and get to know more about preeclampsia, especially for us who are health providers. Yeah, so thank you for coming, and we'll see you at the next one. If you would like to take a selfie, just turn your camera on, and then we will give our best smiles. I'll say 1, 2, 3, and take a picture.
Thank you
So
very
are
much,
we ready?
Isabella. In the meantime, if participants are reminded that we will need a feedback on the program that we've organized, so in the chat, there's a link. If you can give us a feedback on how well we've organized the program in terms of content and form, so that we know how to do better next time, we'll be very grateful. Thank you.
Thank you, everybody, and see you at the next one.
Isa, are you taking the selfies?
Yes.
Remember to move on to the next pages so that everybody can be catch up.
Alrighty.
And if our music, thank you.
All right, done.